Hey Ron,
Hi there.
I've got you now. It's all okay. I
That's
can
great.
see you. How are you doing?
I'm great.
Good.
Nice to talk with you.
Yes, and you. Thank you so much for giving up your time that's so limited just to talk to me in
Well,
my
like,
bed.
well, your limited time is when you're able to do stuff. So
Yes. Yeah,
I appreciate this.
very true. Thank you. Can you see me okay in terms of...
Great.
Okay, brilliant. Um, So yeah, as I think we kind of mentioned, the kind of purpose of these chats is just to be really informal and just to kind of give the community just some updates basically on the lab day to day and basically things which you've found out or things which you've not found out and yeah, just a little bit more specifics for the community, which I think is quite a good idea. So if you're happy to do that, I can.
Yeah, absolutely.
Yeah.
Yeah.
Cool. Okay, so I'm going off the top of my very foggy head. So we've had the symposium a few months ago, which obviously went really well. That was in August now, wasn't it? The community symposium.
That's right.
Yep. And I have to count the months back, 3 months. That's right. And basically, if you can sum up kind of what's been happening since then on a kind of lab day-to-day basis, I think maybe a good place to start might be what's your typical day at Stanford at the Genome Center?
Well, my typical day is usually trying to solve various problems that occur. It's kind of a frustrating kind of thing to feel like, because some of them are scientific problems and some of them are, you know, personal interaction problems.
Okay.
And that's always been the case. It's doing research is very stressful.
Yes.
An awful lot of experiments, some may not work right. The other kind of problems you can find is that you are counting on other people to deliver something correctly and they don't. You know, and it's, for example, you know, we get samples from the blood bank for healthy controls, and then we're finding out that, well, maybe these people aren't healthy, and
Yes.
that's critical.
Yes. Yeah. It's
I think that kind of frustration, and it means, oh gosh, now we got to go do it ourselves, and that adds extra resources and demands on us. And one of the latest big frustrations was the discovery that some of the biobanks that we were going to rely on for samples weren't really collected correctly and stored correctly. And unfortunately, correctly isn't correctly in the standards that they messed up. It has to do with what we find we have to do to make to get valid data.
Yeah.
And that's a problem with the biobanks because they're often not collecting data.
Mm-hmm.
And of course, what we are doing often is collecting new types of data. And then we have to do all the controls to make sure that we're getting the best possible data and reproducibility.
Yeah.
And then we find that, oh, and then we learn how it has to be done. And then we find out, no, the biobanks weren't stored that way. They weren't collected and processed that way. We can't use them.
Yeah.
Very frustrating.
So you're back to stage one.
Yeah, we had to turn down 300 samples.
Okay.
And I don't necessarily blame the individual. It's basically a process problem with biobanking.
Yes.
Because often the biobanks are made, you know, years earlier.
Yes.
Possibly before a particular technology was ever developed.
Okay.
And the hope is, of course, that the biobanks will work fine however they did them, but it's not necessarily the case.
Okay.
So those are kind of the frustrations we get into, and we'll get over it. It's just going to take us a little longer.
Yeah.
But it's far worse to do a lot of studies and publish a lot of studies to find out then that, in fact, the data are all invalid.
Yeah, for sure. Yeah.
Now you spend even more money and time.
Yeah. Yeah.
So you want to find this earlier.
Yeah. So in a way, if we look at this in I guess a positive aspect, at least you found out this earlier rather than later.
Well, and that's exactly how research often goes. That you count on certain things, you count on a certain reagent, you count on a certain company supplying something, and you find out that in fact, oh no, it's we discovered it early enough to backtrack and try to do it correctly. And that's frustrating.
Yeah, yeah.
But that's the frustrations of research, and it's actually far better to figure those out.
Yeah.
So we spent a lot of time trying to make sure to validate and check results, and that's been the history of what we've done. And usually we find that the way it's done in the industry is not right.
Okay.
That's scary.
Yeah, so this is more, it seems like a pervasive problem within the industry as a whole.
Well, and it has to do with people who are so excited to get the results and get it published that they don't think up front to make sure that everything is in fact valid. And that's the pressure to publish. So when people say, even to us, you haven't published yet, you haven't published yet. Well, that's kind of a high pressure. And yes, we could, but you wouldn't, and the results will probably be invalid.
Yes.
And that's not okay.
Yeah, for sure. I think that you've hit on a point actually that might be quite good for some clarification. Obviously, with myself being on the forums, I see kind of quite a lot of things written down, and I don't know if you can clarify your stance on publication for people, because I think sometimes it's been confused somewhat about how publication and OMF and the Genome Centre work, and what your aims are. So I don't know if you can just briefly clarify your stance on the publication process, what's perhaps wrong with it in general.
Well, we feel that the community needs the data that you collect as soon as possible. But what they also need is making sure that the data are in fact valid.
Yes.
And so that's another process. You may have collected it, but if you're not sure that it's right, and it's not necessarily because somebody did something wrong, it has to do with uncovering something in the process that makes you suspicious. And then you have to do a lot more work to make sure that it is in fact valid. And you don't want to release it until you know that that's true.
Yes.
And that's frustrating. And then you have the problem about publication.
Yes.
And I think what we see in the review process of papers and grants is very much like the experience with trolls on the internet.
Yeah. Okay.
I basically now thinking of them as a bunch of trolls reviewers.
Yeah. Yeah.
And there's something psychological. I talked to Janet about this. That when you're allowed to criticize and you have all the power to criticize all you want, and nobody knows who you are, which you see on the internet all the time, people get sometimes incredibly nasty, unnecessarily nasty. And I think that's a problem in society in general.
It's that anonymity factor.
Yes, and I think it's hiding behind, you understand you don't want to have those individuals doing the reviews to be attacked, so that's why you keep them secretive. But it seems to me somebody else needs to overlook that and make sure that they are not being unnecessarily nasty.
Yes.
And in fact, if the criticism in fact are valid.
Yes.
And that's a secondary level, which is what the editor should do and what the grants manager should be doing. And they don't do that.
Yeah.
And I think including, an extremely negative personal attack should be thrown out. That review can count.
Yeah.
That will stop it. Because people will begin to know that if I put in something super harsh that's really a personal attack, then I'm wasting my time.
Yeah.
Anyway, I think we need some fixes on that, and I'm really worried about the young people in science. I mean, I have enough experience that I'd say, oh yeah, there's another negative, there's another troll, and kind of dismiss it. But the young people sometimes, they don't know how to deal with that, and they take it very personally that they're not a good scientist.
Yes.
and that's not good for anybody.
Mm-hmm.
There's a lot of really talented young people out there. It's incredible.
Mm-hmm. Yeah, for sure.
So there's a total secondary thing, but it's people might like to know the frustrations of actually doing the research. Is you have the frustrations of the actual research, and then you have the frustrations of the people looking over your shoulder.
Yes. Yeah, I understand. I mean, one point that comes up, this is a kind of tied in with that, but secondary, is that I think some people have maybe perhaps been confused over the fact that maybe they didn't think that you didn't want to publish, which isn't true, is it? It's not about not wanting to publish. I mean, you can tell me about that.
Well, yeah, I certainly want to publish. It's actually a little, you know, you're going to get heavy criticism, so there's a little bit of intimidation there.
Mm-hmm.
Someone's going to find something wrong, and then you have to start arguing.
Mm-hmm.
But no, why would you do the research if you didn't publish it?
Yeah.
The concern really is the fact: is it really correct?
Yes.
And then and there's some stuff in our severely ill patient data that I am suspicious of. I don't know what's wrong.
Mm-hmm.
I'm just looking at the data. Is this really valid?
Mm-hmm.
And that makes me people, why we're not putting it up.
Yeah.
It's a gene expression stuff, and I look at it compared to our trauma, and it just doesn't seem right.
Okay.
And so I think what we're going to have to do is go back, and the problem is with severely ill, we really can't sample those patients again.
Mm-hmm.
And we'll have to do, and I'm trying to come up with what can we do to see if in fact it is valid. We'll have to do an extra experiment.
Yeah.
And I think it's the processing that we chose to use to try to get better data. I think it may not have done that.
Mm-hmm.
So I think it means we have to develop a different process to validate that data.
But this isn't unusual in science, is it? This finding out that something hasn't worked quite correctly is not an unusual process. And
it's not unusual, and also the problems are with what we're doing is we are always trying to push the envelope.
Mm-hmm.
We're trying to do things that have never been done before.
Yeah.
We're trying to collect data that have never been collected before.
Mm-hmm.
And a lot of research is instrumentation that's commercially available. You take your sample, you put it in the instrument, and then you publish, and you don't even think about it. Mm-hmm. It's up to the manufacturer to make sure that it's valid.
Mm-hmm. Yeah.
That's a lot easier.
Yeah.
So, like a lot of the cytokine work, that is so easy.
Mm-hmm.
There's an instrument that does it. You take the sample, you put it in, you get the data, you're ready to publish.
Yeah.
Right. And you don't question that.
Yeah. So.
Although, it actually could be wrong. But yeah. It's just means that everybody's wrong.
Yes. Yeah. No, that makes sense. I mean, I think when you look at the scope of the severely ill study, and when I first saw it, which was a fair while ago now, I mean, I couldn't believe how many things you were measuring, and the cutting edge technology that was being used to measure it was and is unprecedented, really, in research, but also completely unprecedented in ME CFS patients.
Well, for example, we had 20 patients, and we were looking for evidence of viral infection.
Mm-hmm.
And we were looking for virus particles in the blood.
Mm-hmm. We
didn't find any that were pathogenic viruses.
Yeah.
I was a little surprised at that. I mean, given so many patients say they have HHV6 or HHV7 active infections, no, we couldn't find any evidence of that.
Mm-hmm.
We looked for DNA in the blood, which means it will come from any site. So a lot of people say, well, that doesn't mean anything, but maybe it's not in the blood. But if there's any infection anywhere in the body, it's now a lot of data that said, well, the DNA eventually, for the DNA viruses, will eventually get in the blood.
Mm-hmm. Yeah.
As one of our advisors, Ron Tompkins, says, the blood's the sewer of the body. Everything gets dumped in there.
Yeah, yeah.
And it has to get dumped in there to get rid of it.
Mm-hmm.
And it doesn't just stay in the body.
Mm-hmm.
The body cleans out everything.
Mm-hmm.
So, And that we found a little bit in a couple of patients, but we found more in the healthy controls. So
Yes.
what did that?
Yes, I saw the data. I saw the data on that. That was very interesting.
I'm nervous about that. Is that really right?
Mm-hmm.
Because I know so many patients are worried about having viral infections, and they take antivirals. And now I'm suspicious that the antivirals are doing something else. That's the other thing that people don't. They think all these drugs are absolutely perfectly targeted, and that's all they do.
Yeah.
None of the drugs are perfectly targeted. They do all sorts of other things.
Mm-hmm.
And the difficulty that the pharmaceutical industry has is it has to do well what it's supposed to do, and it's not supposed to do stuff that harms the body significantly to hurt the patient.
Yes.
And that's the game, and that's hard, and they have to keep modifying the drug.
Yeah.
But it's all these things called side effects.
Yeah.
It's that because the drug's doing something else. Well, sometimes what something else is actually beneficial to the patient.
Mm-hmm. Yes. I think I read something. This is going. It's kind of on topic, but slightly off topic about antivirals and Valtrex and Valsite and about the modulation. It was in a paper about the modulation on adenosine as one of the functions of the antivirals, and I kind of thought possibly that could have some effect.
Right.
Yeah.
And sometimes there are quite a large number of extra effects.
Yeah.
And it can be difficult to track. Our technologies are getting sufficiently good that we can actually explore some of them.
Yes.
We have especially we have a patient before and after they take these drugs.
Yes.
And look at what else is being affected.
Which is reassuring that the technology is now catching up.
Yes.
Yeah, because that's been a problem.
Not only that, but we could possibly understand some of the side effects, and then we have an assay for improving the drug.
Mm-hmm.
You can see that it's doing the wrong thing possibly, and we can now have an assay for the wrong thing and for the right thing, and then change the drug and look to see that you're making improvements.
For sure.
Anyway, that's the future.
Yes. Talking about, I've got a kind of little notepad which I've noted some things down. Talking about essays kind of leads us nicely into a little kind of thing I had written down was the nanoneedle progress. So obviously there's been tons of talk about the nanoneedle in terms of community talk, but also at the symposium it was a focus of the nanoneedle, and there's been some developments there, I think, recently, hasn't there?
Yes, and the thing that I'm talking to the community about is that what we're excited about is a simple measurement. It could be done very inexpensively. The instrumentation could be made fairly simple.
Mm-hmm.
It's an assay that could be done in a doctor's office. It takes no more than a drop of blood, and we could probably even do it with much less.
Mm-hmm.
The nanoneedle itself, if mass-produced, would probably cost around $1.
Yes.
So it's not going to be an expensive technology from the point of view of supplies.
Yeah.
And so far, it's been quite reproducible.
Mm-hmm.
Now, that's good news.
Yes.
And we see it in the patients. And it's a very, when we do these measurements, one measurement from a patient involves about 3 billion actual individual electrical measurements.
Mm-hmm.
It's just trivial for the technology that's being used,
Mm-hmm.
but it's not like your standard medical assay, which is one measurement.
Yeah.
So sometimes a single measurement can be wrong and give you a false positive or a false negative.
Yeah.
Also, the people don't recognize in the medical community there's a fairly large number of false positives, false negatives. That's hard to deal with for the patients.
Mm-hmm.
They like to know, for sure.
Yeah.
And unfortunately, that's not available.
No.
So one way to deal with that is try to make sure you have multiple kinds of indications. If you're making a measurement, you have symptoms in addition to the measurement. Doctors tend to take these biochemical measurements as absolute. And there are some assays that, in fact, are probably around 10% false positive, 10% false negatives.
Okay.
And some of it has to do with the level. It just simply can't detect that level. Or it's detecting something else, cross-reacting in some way to give you a false positive. I think the research community with all these new technologies will improve upon that and actually go after why we have false positive, false negatives. That's been a little beyond our capacity in the past. Yes. And now much better tests. So this is just an illustration. Instead of making one electrical measurement, we actually make 3 billion electrical measurements in about a 3-hour period.
Yeah.
So it gives you that doesn't mean it's correct. It just means the electrical measurements are correct.
Yes. Yes. Yeah.
So that comes to the other problem. What are we actually measuring? I mean, yeah, we're measuring impedance. Mm-hmm. But what is biologically going on that we're measuring? We haven't figured that out yet.
No.
And sometimes you never figure it out. But that's one of the side efforts is what in the world are we actually measuring? Yeah. And that will help. And in fact, it could turn out that we're measuring something unexpectedly that nobody knew before was present in the patients. Mm-hmm. And in fact, there is a much better way to measure that. Mm-hmm. That's a real possibility.
Also, in terms of the
what's it?
I was going to say carry on. If I was going to say in terms of the nanoneedle itself, I know there's been some developments in terms of you've managed to make it. Is it reusable currently? Like you can now reuse it?
Yeah, they figured out a way to clean it. The nanoneedle itself is silicon oxide, which is sand.
Mm-hmm. Yeah.
Which a lot of electronics are. It's a very good resistor in terms of it doesn't allow insulator, doesn't really allow electrical signal to pass. Mm-hmm. And the other is gold, which is pretty inert. Mm-hmm. Of course, it has very tiny amounts of gold. Mm-hmm. But still, we have to clean off stuff, and there are some really aggressive ways to clean in the electronic industry. Mm-hmm. But those really aggressive ones are etching the surfaces too much and degrading. So these things are not very thick. They're nanometers in thickness. Mm-hmm. So we're at the molecular level with these things, and it makes it easy to damage it. So either it doesn't clean it adequately, or it damages it with a more aggressive one.
This may sound a very amateur question that I've just thought of, but because the cost of the nano needle test is so low at 1 cent, did you say?
No, it's about $1 probably to make these.
Okay, if it's at $1, can I just can I ask what's the benefit in having it be reusable when the cost is already so low, if that makes sense?
Well, that's not what we have to pay because it's not mass produced.
Okay.
So if we went to China and said we would like to have 10 million of these or 100 million of these, yeah, that's when we get to $1.
Okay, yeah, makes sense.
So it's not the materials cost in this, it's time.
Yeah.
And this has to be made by Raheem, mm-hmm. who's a high-level electrical engineer.
Yeah.
And it takes him a couple of weeks to make a batch of them.
Okay.
That's a lot of money.
Yeah. Yes. No. Yeah.
What's worse is when he's making the chips, he's not doing the experiments.
Yeah.
We tried to find other people to make them, but it's just tricky to do some of this stuff. Mm-hmm. And he's getting very frustrated because he's gotten some people who can make them okay, and other people that they don't come out and work very well. There's a lot. He can test them, and then a lot of the chips are actually defective.
Yeah.
And so that's a real problem. Okay. So he's gotten too frustrated about that and decided he was just going to make it himself, which is why he was so motivated to clean them.
Yeah.
He doesn't have to go back and make more.
makes perfect sense now. I was thinking in terms of it costing $1 each time and fairly easy to make, but obviously that's clearly not the case. So yeah, okay. Um,
He's working on another technology, but we haven't gotten it to work yet for this purpose. But we have um, an intern that's come in who has a BS degree in electrical engineering. He's really good, and he's trying to make a different device work that's easier to fabricate and cheaper. So this new device is about a cent to make,
okay.
but it doesn't require mass production.
Okay.
So this is using an inkjet printer. So it's a little larger, but that we don't need it so small. It still will work with a drop of blood.
Yes.
But the advantage of that is that anybody can make it, and so that would make it easy to export.
Yes.
The only thing would not be so easy is the electronics data collection part.
Yeah.
But almost anybody could fabricate that as well.
Yeah. I mean, that's an exciting development.
So this happened yesterday, so I try to get people updates, current updates. So yesterday,
Yeah.
A patient came in that seemed pretty okay. Didn't seem you can kind of tell when a person is struggling and a little bit fatigued, and especially coming, driving or having somebody drive them to the center and walking in and getting there for the blood draw. This person seemed pretty much at the high end. And when we made the measurements, her signals were very low.
Okay.
And that made us nervous that, well, here's the first person we've seen that appears to have chronic fatigue syndrome, but that's getting a pretty low signal. And we find that she's on all sorts of drugs.
Okay.
And that's actually a little encouraging. Because, and we have to explore this a little more, and that's because she looks pretty good, and she's on all sorts of drugs, and maybe that's modulating the disease.
Yeah.
But what is it that she's on?
Yeah.
And is there and we need to find maybe another patient that's on some of those drugs. Maybe it's actually helping.
Yeah.
And we'll have to explore that more. So this is kind of bad news that it's not quite clear how you would diagnose this person from the electronics.
No.
It's above healthy controls, but only slightly.
That's really interesting.
Yeah. So that will be and she's local, so we'll be able to see her again if she's willing to divulge the drugs and so forth. But these are what I call accidental discoveries.
Yeah.
And that happens a lot in science. And you just have to be aware of it when it happens.
Yes.
And that's why it's very important not to be looking for a particular thing.
Yeah.
Always keep your eye open. So, for example, in this test, we might have said, "Oh, she looks okay. I bet she doesn't have chronic fatigue syndrome. Let's throw out the data."
Yeah.
That can happen.
Yeah.
That's when someone's looking for a result. You never ever should do that.
Yeah. The negative results.
And that's to some extent what happened to the PACE trial.
Yeah. Yeah.
They were really believing in their hypothesis, and they did all sorts of things that made it look more like the hypothesis was correct, without really realizing that they were manipulating the data.
Yeah.
And what you get with that is the wrong conclusion.
Yeah, absolutely. Absolutely.
That's not how you do science. You don't try to prove something is right. You ask the question, what is happening?
Yeah.
And when you get something that's unexpected, actually that's it's a potential accidental discovery that could lead you to the right answer.
Yeah.
You always pay attention to those.
Yeah, the value of the null result or the negative result can be just as,
And those can happen to some of the smartest people, and they can also happen to some of the less talented people.
Mm-hmm.
And which is why you really like to have lots of people working on this problem.
Yes.
Because these accidental discoveries are very, very important. The rituximab
Mm-hmm.
was an accidental discovery.
Yeah.
Now, that could have happened with the oncologist, Flugamella, working on this,
Mm-hmm.
saying when the patient said, my chronic fatigue syndrome symptoms have improved, they could have said, well, that's not our business.
Yeah.
We're oncologists. We're trying to cure your cancer.
Yeah.
That's irrelevant.
Yeah.
And ignore it.
Mm-hmm. But
And I suspect that actually did happen. If you think about lymphoma, is a fairly common cancer in chronic fatigue syndrome patients for some reason, it appears. But it's a fairly common cancer anyway.
Mm-hmm.
Given how many patients have chronic fatigue syndrome, there must have been a number of lymphoma patients with chronic fatigue syndrome. And rituximab is the drug of choice.
Mm-hmm.
I can't believe that it hasn't happened many, many times.
Yeah.
In all those other cases, it was ignored.
Yeah.
Now, or the patient didn't say anything.
Mm-hmm. Yeah, for sure. It's an interesting thought, and I agree with you. It's probably
very important for doctors and researchers to pay attention.
Yeah.
And especially pay attention to something that doesn't make sense.
Mm-hmm.
And to them, it could say, well, I have no idea why rituximab would make your chronic fatigue syndrome symptoms better. That makes no sense.
Mm-hmm. Let's look into it.
And then not stop there, but go ahead. And that's why I give Flugamella a great deal of credit, because they didn't stop there.
Yeah, for sure.
But that's also why you want lots of people making observations.
Yeah.
And that's why we need to recruit more people in to doing this work.
Yes.
We shouldn't be trying to sequester all the funds and only a few people, chosen people, get to do this.
Yes.
That's not the right way.
No. That's the whole philosophy, isn't it? The open approach to
Right.
working with decent researchers. I do know, as another point, that I think, correct me if I'm wrong, because I may well be wrong on this, but some of the funding from OMF has gone towards a full-time, is it researcher at the Genome Centre, or a full-time member of staff? I don't know if that's correct or not. I believe I thought the Stanford Genome Centre had taken on a new member of staff recently.
Well, one of them is a bioinformatics.
Yes.
One of the slow points we have is that Wenzong has been helping us,
Mm-hmm.
but he has another full-time job.
Yeah.
And so and he's gotten some people that help him and so forth, but it's not enough capacity. And some of that stuff can be done, he's at Harvard.
Yeah.
Can be done at some other institution, but it's really great to have somebody in-house where you can talk to on a daily basis and say, hey, would you look at this or look at that? And it's more difficult when they're far away. So
the bioinformatics is quite new.
A full-time person to be dedicated to this project. In the past, we borrowed some help from other people because we didn't have enough funds. So that's a tremendous help. That will speed things up.
Yes. Okay. Brilliant. That sounds great. I mean, I'm just trying to look through in terms of some exciting news. I think the newsletter has just gone out on the Phoenix Rising forum, and it will go out worldwide. So the OMF newsletter that's gone out, but Jonas Bergquist has joined the scientific advisory board, which was
and that's great. He gave a really nice talk.
Mm-hmm.
Some humorous comments in there as well in his talk.
Yeah.
And we have worked with him, and we have done some not high-level funding, but some low-level funding of his. for his research that he needed. He's got a really nicely equipped laboratory, which is great. He, he's very involved in lots of things. So it's really great that he spent some time on this. And he's particularly good at the spinal taps.
Yes.
And storing spinal fluid. And one of the keys when I was talking with him, and I was asking him about the spinal taps because they're often painful, and that makes me a little leery to try to get a patient to do it. And he said, no, they don't mind at all. In fact, they would rather have a spinal tap than a blood draw. That's a big, and blood draws are really nothing.
Yeah.
And to most patients, the blood draw is not a big deal.
Yeah.
And he said you just have to do it correctly.
Yeah.
So he must be very skilled.
And it's an area which I think I call well, I may be wrong on this, but from the literature I've read, I don't think it's been explored very much at all because of those very problems.
Because of those issues, right? And you often have to go to a special facility, so there's a delay in getting the sample. I think there's probably very little delay in his case.
Yeah.
And so it is an important area to explore, and of course that's what we will probably be trying to get his help on, is exploring those kinds of areas.
Yes. Because I was reading earlier.
And of course, he has an awful lot of the equipment and stuff, so he can himself do two things. What he really needs is to pay for the detections time and stuff like that, and reagents and so forth. So it's a matter of getting together with him and talking about what is it that we should be doing.
Yes. His field of expertise seems to be quite broad, but from the gist of it that I was reading, it was mainly kind of neuroimmunology and various related.
He's effectively an analytical chemist.
Yeah. Okay.
And there's a lot of training for an analytical chemist about how to do an experiment correctly.
Well, that's good.
And this is about false positives, false negatives, and all your quality controls, and an analytical chemist gets that in spades, and in terms of drummed into them. So you can almost always trust an analytical chemist to get good quality data.
Yeah, that's reassuring.
And that's what we need.
Yeah.
We need people that will generate the data that in fact we can believe.
Yeah. Okay. Can you still see me okay, Ron? Because I know it's in the evening in the UK, so we have very...
I just worry about, I have this backlight that I'm trying to make sure it doesn't flood the screen.
No, you're okay, as long as I'm okay, because of the...
We have morning here.
Yes, and it's the UK, and it's not raining, but it's very dark, so as long as you can see me okay, so I'm not just a hat, then that's fine.
I remember that hat.
Yeah. It's to hide the messy hair. That's why.
That's so you don't have to get haircuts. So.
Yeah, exactly. Another thing was the follow up on the T cell with Mark Davis. Now I know that's pretty. It was very preliminary and very new. I mean, that was only a few months ago, wasn't it? Really. I don't know if Mark's talk was fantastic. I think a lot of the community saw it and were really encouraged by it. I don't know if there's been any developments there that can be shared, or if there's been any news or anything, or what's...
I think what's mainly has to happen is we have to collect a lot more data. And that's in process. A little bit of the new things are that there are now 4 postdocs that work for Mark Davis that are now in the Genome Center. Okay. So his labs are largely are in the Beckman Center on campus next to the medical school complex. He has an immune monitoring core that does things like cytokines. The recent paper by Jose Montoya was done in the Immune Monitoring Corps.
Okay.
That's in a building that's adjacent to the Genome Center.
Okay.
So we're still at Stanford campus. It's just that we're a bit on the opposite side of campus from the medical center.
Yes.
So we're about 5 to 10 minutes away by car, and you could almost walk there.
Yeah.
The biggest time is finding a place to park.
Yeah. Yes.
It's a bus service, and so if you're organized, it's a lot easier. But if you're disorganized and have to do it at the spur of the moment, then you have to drive or bicycle.
Yes. Yes.
So we're hoping to have more interaction, and we're also a postdoctoral fellow that works for Lars Steinmetz, who's in the Genome Center. He's a mitochondrial expert.
Okay.
Has a postdoc that has developed some of the methods for single cell sequencing, and we will get those sequences from single cells, and also RNA levels from single cells.
Mm-hmm.
RNA levels will help tell us a little bit about something about the nature of that particular cell.
Mm-hmm.
So that will start as soon as we have the funds for that. We wrote after getting turned down from the cooperative centers. That was I was really disappointed in that, because we put together a large group of really talented people.
Mm-hmm.
And most of them only peripherally connected or not connected at all to chronic fatigue syndrome.
Yes.
And the problem is that the problem with really good people is they have no difficulty in finding things to do.
Yeah.
So they'll go away and do something else.
Yeah.
So I'm going to try to keep them committed. And so we wrote another grant immediately after learning we were not awarded.
Yeah.
Mostly putting in Mark stuff.
Mm-hmm.
It was an R1 grant, so it has a financial limit much smaller than the cooperative center, so we can't put everything into it.
Mm-hmm.
But now it's all immunology, so we sent that to the Allergy and Infectious Disease Institute at NIH.
Okay.
And Mark has a good track record in that institute for grant funding.
Okay.
So there's a chance that we'll get that project funded. And then we're also going to try to get donations from Open Medicine Foundation to support that research as well. And that's in the works.
Yes.
Because that's a really important thing. And we got really the best people doing those studies, and it would be a real shame not to really carry it out.
Yes. Yeah, for sure. I mean, that brings me on to kind of another point that I had written down, which if you can just kind of describe OMF as opposed to Stanford Genome Technology Center in terms of pretty much OMF provides the funding for the project.
Right. So OMF is a very small scale NIH, but very targeted about what they raise money,
mm-hmm.
Not from taxation, unfortunately, so it would be forced to give.
Yeah.
But by people donating money, and then they funnel it into doing research,
mm-hmm.
And that's why we have a scientific advisory board
mm-hmm.
To try to think about what research should actually be done.
Mm-hmm.
Now I've put a lot of people on the scientific advisory board that I'm hoping that would be good researchers.
Yeah.
So I don't think it's a conflict of interest, because that was deliberate. Like Mark Davis is on the advisory board, because I want him engaged, and this is one way to get people engaged.
Yes.
Is to have them think about the research and talk about it, what's going on,
mm-hmm.
and get them interested.
Yeah.
So and Bob Nabio is on the advisory board.
Yeah.
He's a whole different field from Mark Davis and so forth. And then there are other people on it, like Jim Watson and Paul Berg, that don't have labs.
Yeah.
But they are just using their own wisdom, experience from the past to help make decisions.
Yeah.
So that way we don't just fritter the money away in tiny little projects.
No.
It gets very heavily focused on the right things.
Yeah.
And it's not what someone proposes to do. It's actually very targeted. This is what needs to get done.
Yes. Yeah.
So with Bob Navio, he really needed to do a validation study.
Mm-hmm.
If we're going to, and that's important. We need to know, is that reproducible, or was that somewhat of an accident?
Mm-hmm.
And it's also possible that it is a good indicator, but not quite as good an indicator as the original study.
Yes.
But it tells us a lot anyway, and that's why you do the valid. How valid is that initial study?
Yes.
And we actually initiated that before it was published.
Okay.
Because we knew about the work.
Yes.
So invited him also on the advisory board, and because he's one of the state of the art in metabolomics.
Yeah.
And mitochondria diseases. So he's a perfect person for this.
Yeah, absolutely. Yeah.
So we funded that.
That was OMF funding the validation study.
Yes.
Yeah.
That's still ongoing. He has a small lab,
Mm-hmm.
so it takes longer than maybe a big lab would take. But I think it will be done correctly.
Yes. That's really important. Just a couple more things. I know you haven't got much more time. I know there was Mike Snyder has been doing some family genetic
Yes.
stuff.
So Mike Snyder is another person to get involved in this, and that's a little easier because he's also in the same building
Okay.
that the Stanford Genome Technology Center is. We are on the first floor, and his labs are on the second floor.
Okay.
And we share a lot of facilities.
Yeah.
And there's a lot of cooperation between the two. He was a past postdoc of mine.
Mm-hmm. Yeah.
But he doesn't. He listens to me, but he doesn't necessarily chow to my ideas.
Okay. Yeah.
He's chair of genetics, and he's done a superb job at chairing genetics.
Mm-hmm.
Stanford Genetics Department now has been viewed as the top genetics department in the world, and for the last, I think, 6 years in a row.
Wow.
And I believe that what they're all doing, they're doing the leading edge of so many things in genetics.
Mm-hmm.
So he has an office in our building, and his office is right next door to mine.
Okay.
So and he has good people, and has a lot of bioinformatic kinds of developments. He's got a large lab.
Mm-hmm.
So we're trying to get him hooked into doing some of this.
And he's working on the family in terms of familial genetic in
Right.
CFS.
Right.
Mm-hmm.
And that's been interesting in the sense that what you often see in clinical studies is you talk about patients and then you have controls, and they make a point that the controls are totally unrelated to the patients.
Mm-hmm. Yeah.
What Freshta has been doing is just the opposite. Her controls are family members that are healthy.
Mm-hmm.
And what you get out of that is that they are, especially if they're living in the same house, certainly you have the same background, but you also, if they're in the same house, you have the same environment.
Yes.
Probably the same food.
Yes.
Yeah. Share, probably depends on how closely related they are, but they share a fair amount of the genetics.
Yeah.
Well, that gets a lot of the noise,
Mm-hmm.
and that's what you're finding.
You can control some of the condition.
When you compare and then you collectively do that with all the different families,
Mm-hmm.
and you can see more, she says now, with using that style.
Mm-hmm. Okay.
And that's just because when you take some unrelated person with an unrelated environment, so unrelated genetics, basically that creates what you would call noise.
Yes.
And making it more difficult to see what's going on.
Yeah, for sure. I guess just before we wrap up, just a couple of things. Suramin, everyone's talking about Suramin, and obviously there's been problems in terms of it's not very easy to get your hands on, so to speak, as a researcher, as far as I know.
This comes from Bob Navio, because he's done some exploring.
Mm-hmm.
If you look in like Wikipedia, and you look in do some searches on the web,
Mm-hmm.
And you look up side effects of Suramin,
Mm-hmm.
They're pretty scary.
Mm-hmm. Yeah.
But Bob Navio knows that that's not really true.
Mm-hmm.
And so there's two issues with this. One is that the amount of Suramin you use clinically for sleeping sickness is lower.
Mm-hmm.
The amount he uses is also lower.
Mm-hmm.
What's happening in the bulk of the literature is that the CDC did a big clinical trial of Suramin for cancer.
Mm-hmm.
And what happens, I'm guessing on this, but I think what usually happens in cancer is that you try to use a highest dose that you can. And I think anybody with cancer knows, the chemotherapy, it makes them sick. Well, partly they're using as high a concentration as the patient will tolerate,
mm-hmm.
in the hopes that it will have a bigger effect.
Mm-hmm.
That's what they did in the case of Suramin, I think, is they used the highest dose that the patient could tolerate, which basically meant all sorts of side effects.
Yeah.
And so those are the side effects you see.
Yeah.
That are reported now.
Yeah.
But and that's what I thought was the answer. And then Bob says, actually, no. There's something else going on, maybe. And that is that when the CDC did that, they did not get their Suramin from Bayer, who's the major supplier of Suramin for clinical use.
Mm-hmm.
I don't know why. It may have been financial. It may have been Bayer didn't want to participate. They contracted out the Suramin to be made.
Yeah.
And Bob thinks it had a number of contaminants.
Okay.
And he knows that from his mass spec.
Yeah. Yeah. The tools of the trade.
And the Suramin he says, from Bayer is very clean. The Suramin probably that the CDC used is not.
Mm-hmm.
But they made a large amount of it. And he said that then is being sold off to suppliers for non-clinical use.
Okay.
So if you buy Suramin on the market, you're possibly getting the old CDC Suramin. It's not necessarily newly synthesized.
Yeah.
And so that Suramin might have problems. And we're concerned about that because we're trying to do tests and so forth in the lab. I want the clinical version.
Yeah.
Because if there's contaminants, we might then see problems with those contaminants.
Yes.
And so that's what I've been struggling to get access to some clinical grade Suramin.
Mm-hmm. Mm-hmm. I remember reading a lot of the side effects as well in the clinical studies. I think it was for HIV and stuff. Was the dosage they used was massive compared to what Bob has been using for autism, for example.
And the reason they did the Suramin trial, I think, on HIV was the fact that it decreased infectivity, if I remember correctly.
Mm-hmm. Yeah.
There's some process in the infectivity.
Mm-hmm.
But they were trying Suramin by itself in those studies,
mm-hmm.
and it wasn't very effective. Well, that doesn't mean it won't be extremely effective in combination.
Yes. Yeah.
So there is reason to actually reinvestigate that.
Yes.
In combination with the others. So if you can block infectivity, and then you block replication, and you can block, all these other different processes, it may even be more effective, and then also you may not have to use high concentrations.
Yes. Yes. And it was at the high concentrations, I think, that the adrenal suppression and other effects like that were reported.
I think, yes. So that's another thing that the patients have to understand is it's not about the drug, it's at what concentration are you talking about?
Yeah, exactly. Yeah.
And basically, if you take anything at extremely high concentration, including water,
Yeah.
lethal.
Yeah, for sure. Okay. I guess just to wrap up, because I know you've got to go. Can you give, I guess, the community a kind of maybe overview of what might be happening up until Christmas? I mean, you obviously this is a day-by-day process, so you encounter, as you've said, um, unusual situations and new things happen each day. But what are the plans up until Christmas? I guess for you that you'll be doing.
Well, I think one thing we need to do is to start some very small-scale clinical trials.
Mm-hmm.
And the question is, what did we try? And that's what we're kind of trying to sort out. And some of that is also talking to drug companies. Is there anything they have that might actually be effective?
Mm-hmm.
Some of those are actually themselves in clinical trials. Is there any drug that's going to be, through phase 3? It might be available.
Yeah.
And I'm trying to make a collection of those because there's a lot going on.
Mm-hmm.
And that includes all over the world. So I have trying to make lots of contacts with drug companies. Yeah. We have a lot of startup drug companies in the Bay Area here, so I talk to them. There are some candidates that ought to be considered. The question is, when can we actually try something?
Yes. Yeah.
And some of them are quite interesting because they have very little in the way of side effects.
Mm-hmm.
But that's something I'd like to, and what I see, it's like a pilot clinical trial.
Mm-hmm.
And it would be something like 5 to 10 patients in that range.
Mm-hmm.
I don't know how acceptable that will be to publication because it's not your typical clinical trial of 100 or 1,000.
No.
But something we could afford. And what we're looking for here is not some small effect, but we're looking for some big effect.
Mm-hmm. Yeah.
And if it's a big effect, you should be able to see it in 10 patients.
Yeah.
Now, there is heterogeneity potentially for this disease. I don't know for sure if that's true. Because there's a lot of heterogeneity in the genetics.
Mm-hmm.
And so that could cause a lot of heterogeneity in symptoms.
Mm-hmm.
And so you can't just rely on looking at the heterogeneity in symptoms and say, oh, it's a bunch of different diseases.
Mm-hmm.
It's more likely, actually, I think, that it's some central control circuit in the body that's gotten stuck somewhere.
Mm-hmm. Yeah.
And because of that, and I think that there may be a lot of sensitivity to the genetics because of the metabolomics, the hypometabolic state.
Mm-hmm.
So patients have much lower levels of metabolites.
Yeah.
That itself is going to create some problems. And then if you're superimposed on that some genetics, we might get a fair amount of heterogeneity.
Yeah.
So I'm aware that it might be a very heterogeneous disease,
Mm-hmm.
but it also might not be.
Mm-hmm.
So given that almost none of these drugs really, in the past, because people are taking lots of stuff, helps very much,
Mm-hmm.
but we might be able to see something in a small clinical trial.
Yes.
And there are quite a few donors that have an affected person, a loved one, and they are, some of them are much more connected to doing a clinical trial because they realize that is much closer to helping.
Yeah.
And of course, it will be expensive, but they're willing to put that much money out with taking a risk,
Mm-hmm.
as opposed to research, which means maybe in 5 years I'll have something for my loved one.
Yes.
They would like something more immediate.
Yeah.
So I think we might be able to get a donation for a clinical trial where that donation would not come in for research.
Okay, that's interesting.
So I don't want to compete. I don't want them to be competing, if possible.
Yeah.
Because the clinical trial stuff is kind of expensive.
Yeah, for sure.
And if you do a full-fledged clinical trial, it's incredibly expensive.
Yes. Yeah.
Now, there are certainly individuals that can afford a big clinical trial.
Yes.
Hopefully, it's not needed.
No. Okay. Well, I know you've got to pretty much go now. I know how busy you are.
Go to. Yeah.
Thank you so much.
Nice to chat with you.
Yeah, lovely to chat to you, and thank you so much for your time, Ron. I know how busy you are, and I'm sure the community will really appreciate this.
Fair enough. Glad to do it.
Cool. Okay, I'll speak to you soon, hopefully, and hopefully you can have a stress-free day, somewhat.
Thank you very much. It's unlikely.
Okay, you take care, Ron. I'll speak to you soon. Take care.
Bye. Bye.
Bye.